and welcome to the DNA 2.0 webinar on CRISPR-Cas9 technologies. I'm Laura Whitman, Director of Biotech Marketing for DNA 2.0, and I'll be your facilitator for this webinar. Before marketing, I was a research biochemist in both academic and pharmaceutical labs. We'll start in just a minute, but first a few housekeeping items. 1. Feel free to ask questions throughout the webinar. We will do our best to answer when possible or at the end of the webinar. You can submit your questions by typing them into the question box on the right side of your screen. If your question is not answered during the webinar, we will email you an answer within the next 2 days. Second, we are recording this webinar, and it will be available for viewing on our website in about 48 hours. You should receive an email with the link. I'd like to thank you for joining us and hundreds of researchers from around the world to listen to this webinar. At this time, I'd like to introduce our speaker, Dr. Kedar Patel. Dr. Patel is a research scientist with significant experience in the field of synthetic biology. He is currently developing DNA assembly and strain engineering technologies at DNA 2.0.
Thank you, Laura. This is Kedar. Hello, everyone. I'm going to walk you through some of the things we've developed around CRISPR-Cas9 here at DNA 2.0. So here's a brief agenda. First, I'll give you an overview of the CRISPR-Cas9 technology and some of its applications. I'll tell you about CRISPR products that we have and the different hosts that we've developed CRISPR products for. I'll show you our gRNA design tool to help select targets. We'll give you some info on technology developed here to minimize off-target effects, which is a concern around this technology. I'll briefly describe some emerging applications that we've developed some tools for, namely guide RNA libraries and methods for lentiviral delivery. Okay, so a little background on CRISPR-Cas9. The way this system works is there's a 20 base pair target region which helps target a site on a genome, and this is part of a guide RNA which has a constant region and then this 20 base pair region. In addition, there's a Cas9 protein which is driven by some promoter, which depends on the host in which you're working. The guide RNA guides the Cas9 protein to a site on the genome, and this Cas9 protein then goes and puts a double-stranded cut in the genome. Cas9 has been engineered, so there's versions of Cas9 that only nick DNA, not cut double-stranded. And there's also a version which does not nick or cut, it simply binds to the DNA. The way this works is the Cas9 and the guide RNA pair get guided to that 20 base pair sequence or the complementary sequence on the genome. Immediately adjacent to that 20 base pair sequence is 3 bases, in this case NGG, which is called a PAM sequence. That is the only constraint on the system. You can target any 20 base pairs as long as it's followed immediately by NGG PAM. Because the Cas9 we're using is from S. pyogenes, and other bacterial Cas9s have different PAM sequences. So this method can be, this combination of Cas9 and the guide RNA can be used for genome editing, which means one can knock in genes or replace genes or knock out genes this way. So it's very convenient and very fast. So how does it work at a more basic level? At a more basic level, here you can see that the genome is shown here in the blue track. And by putting a double-stranded break in the genome, one can then repair it in 3 different ways. One here is shown, it's called non-homologous end joining, where the gene is, where the 2 pieces of DNA simply come together and bridge the gap, and a small mutation is created, 1 or 2 base pair, which is a frame shift. So by doing this, you can make a frame shift, so knock out a gene very simply. You have to only supply the Cas9 and a guide RNA for a target sequence to do this. Another way to fix a double-stranded break is by supplying a short oligo sequence. So in this example here, if you can put in a small oligo that bridges the site that was cut and introduces a small change like adding 3 base pairs or a different amino acid or putting a point mutation, things like that, this is a great way to do that. So in this case, you would supply the guide RNA along with a donor cassette. A third thing which is convenient using this method is by supplying a cassette that allows for replacement of a gene. So you can not only knock out a gene, but also replace it with a different gene here shown, with like maybe a 1 or 2 kb sequence where it has upstream and downstream homology, along with a new gene to replace the existing wild-type gene. So lots of these things can be done. It's very convenient and very fast, and can be done in a marker-less manner, which makes it very attractive. So DNA 2.0, to enable this, has developed a bunch of different tools, including all-in-one vectors. What I mean by all-in-one is that all the components necessary for making the double-stranded break at your site of interest can be done with one single vector, which is very convenient. Along with being an all-in-one vector, we enable the researcher to have a lot of choices around the different promoters and things like that. Here are some of the choices we offer. So the guide RNA in this case is driven by a U6 promoter. We have Cas9 driven by 3 different promoters. CMV is a strong promoter, and we also offer 2 other promoters. And on the right here, I show a graph where we validated expression of Cas9 and demonstrated double cutting at the genome level. So this graph shows indel frequency, which is basically a measure of cutting at the genome. So you can see that in this example, if you get cutting at the genome and there's a small frame shift or a small deletion, insertion, point mutation, that will show up as an indel compared to a wild-type sequence. In this example, with the 3 different promoters, you can see that with the strong promoter, we got high efficiency of cutting at the target site, demonstrated by a high indel frequency. And we got cutting even with the other 2 promoters. The idea being that if you can modulate on-target activity, you can also perhaps control the trade-off with off-target activity. If you get lower on-target activity, presumably that might mean slightly lower off-target activity as well. So you can choose a promoter that works well for you. In addition to promoters, we also offer our IP-free fluorescent protein reporters, which means you can monitor expression of Cas9 in the cells of interest. So here you can see we have GFP and RFP, which also are compatible with each other. So in case you end up using a 2 plasmid system, you can use one with the GFP, one with the RFP, and target 2 separate sites or other such applications using these plasmids. So this choice of promoters, as I mentioned, enables you to control sort of trade-off between on-target effects and efficiency versus off-target effects. Here I show also another graph where we compared our results with 4 different targets versus published literature. These are the same targets, and you can see that with our vectors, the results are comparable with published literature. So our vectors have been validated and they're ready for purchase. Now, as I mentioned, one concern around CRISPR-Cas9 has been specificity. What I mean by that is cutting at sites that were not intended to be cut, non-exact matches. So if the researcher was targeting 20 base pair, a 20 base pair sequence, these guys here in the Jeffrey Sander group showed that you can get cutting at any 1 base mismatch in the sequence. So if you look here, this is the 20 base pair target sequence, and immediately following this would be the 3 base NGG PAM. You can see that by varying any of the bases shown with the gray boxes, any 1 base, all the way up to the base right before the PAM, you still see cutting. So this is again indel frequency. This is for the wild type where it actually matches the sequence 100%. And for every base variation from that, you still see cutting. There's more promiscuity at the first 8 bases of the target as compared to the last 12 of the target, the 12 being closest to the PAM. That was one trend they noticed, but again, it's all target specific. Certain targets had more, behave differently in terms of where the mismatches mattered most. One more alarming thing was, here you can see that they varied up to 5 base pairs from the expected sequence for cutting, and they still saw a little bit of cutting. So that was concerning, where you might target a sequence, but even another sequence that exists on the genome that is as much as 5 bases different would still get cut a little. So off-target activity can be a serious problem. Depending on your application, it may or may not be a concern, but it certainly is something to be accounted for. One way to address off-target activity is to use this dual Nikkase strategy. What I mean by that is, as I mentioned, there's a Cas9 that's been engineered to cut at only 1 genomic site, at only 1 strand of the genomic site. So it does not put a double-stranded cut in the DNA, it simply puts a nick. So if you find 2 sites on opposite strands that are close by to each other and use the Cas9 nickase, one would get two cuts that are offset but close enough so that the DNA would come apart and behave as a double-stranded break. So that's one strategy that's been developed, and it's been suggested that that would have 50 or 1,000-fold lower off-target activity. What we've done here at DNA 2.0 is enable you to use a single vector and target two different sites using this nickase to hopefully minimize off-target activity. And it still retains the convenience of having an all-in-one vector. On the right here, I show a graph again of indel frequency, which is a measure of double-stranded cutting and getting mutations. And you can see here, if you compare our all-in-one vector at one genomic site versus a two-vector system, which would use two separate plasmids, and we also compared that same two separate plasmid system. You can see that with the NickaseNinja, where you have two guides on a single plasmid, the efficiency is higher, presumably because there's no co-transformation. You have a single plasmid going to the cells. If you do choose to use the co-transformation method, we do offer the conventional system as well, and we do offer it with GFP and RFP, so you can monitor expression of both plasmids individually in the cells. So this can be convenient for many researchers, and we have a patent pending on this NickaseNinja vector. Now I'll just show you how you can also minimize off-target activities by just selecting the right target. We have developed a tool to help you select a target to hit any site of interest. It also accounts for any other sites that are present in the genome that may be similar to it. It may be 1, 2, 3 bases off, but we give a negative score to those target sites, and these get rank ordered based on ones that are most unique in the genome and least likely to have off-target activity. Let me go to the tool and demo it for you really quickly. So on our homepage, this is dna2.0.com. We have CRISPR-Cas9 listed under products. You can also get to it here under products. We'll go to CRISPR-Cas9. In there, you'll see we have a link to the gRNA design tool. Now there's two scenarios here. If you already have your target gRNA and you simply want to order that guide in one of our vectors, you can just say I have my own gRNA and paste them here in FASTA format. If you want to design it using our tool, we have 3 different ways of targeting a sequence. First, you have to choose which host organism you're working in. Right now, we have products in mammalian cells, so human and mouse, or Saccharomyces cerevisiae, yeast. We have more in the works, and I'll talk about that in a few minutes. But for now, these 3 genomes have been loaded. The PAM, again, we use the S-Biogene's Cas9, so it works well with NGG PAMs. It has been shown to work with NAG as well, although not as well as with the NGG PAM. So if you need more flexibility around finding a target sequence that will work, and you're flexible on which PAM you use, you can select NGG or NAG. But the NGG PAM alone is more efficient, so this is what is our default. If you want to work with a wild-type Cas9 and put a double-stranded cut at one single site, you can select that. If you want to use our paired nickase strategy, where you find offset guides on 2 different strands, you can use our nickase plasmids. So again, we recommend the nickase because it's expected to have less off-target activity and it still demonstrates very good on-target mutagenesis. So we'll use the nickase. This offset just means the distance between the guides. Now, anytime if you're not familiar with these these input boxes, you can hover over the question marks and it'll tell you what the different selections mean. As I mentioned, once you have the genome selected, we'll go with human and the NGG PAM with the nickase. Once you select the genome of interest and you select the guide RNA offset, you can enter your you can input your region of interest in 3 different ways. You can target a gene, which means you can type in the gene name and it's an autocomplete field. You can type in a genomic region of interest. In the human genome, you can choose which chromosome you want to target and then start an N base. We have a 10 kilobase limit, so 10 kb is the limit on the window. The third way you can target sequences is just by simply entering a sequence of interest. This is most useful if you have introduced heterologous sequence into the genome, such as an antibiotic marker or a GFP or something like that. And now you want to target that sequence in the context of a certain genome of interest. So targeting a gene is a common example. I'll walk through that, where one is putting, let's say one is trying to knock out a gene of interest. If you start to type the gene name, it will simply autocomplete your options. So with FOXP2, for example. If you want to knock out a gene, it's best to introduce a frame shift near the beginning of the gene. So targeting the first exon makes sense. So if you check that, the tool will only find sites within the first exon. The idea being if you put in a frame shift near the 5' of the gene, you will produce a very small peptide which should not be functional usually. Once you select the gene, you can search, and the results you will notice are almost instantaneous. In a couple seconds, you see the results. What the results are showing here is a few different things. One is it's showing all the exons for all the different splice variants for this gene. So you see here an exon number is listed with this little gray number here on the side of the exon. The exons are these blue arrow type tracks. And in this case, there were 8 different splice variants. And in each case, it tried to target exon 1 or closest to the 5'. If you hover over the targets, usually you'll see 3 or more targets that are auto-selected, and then up to 100 different targets in all. So I'm back here, I'll just click again, and you'll see, in this case, there was only 1 target found, and it's showing you where it hits the gene of interest. This is again when we're using the Nickase, so you're seeing 2 offset targets, and they are about, in this case, 5 bases apart, and they will cut the DNA and you get a double-stranded break in this case, and this one hits all 8 splice variants of 8 possible ones. Now if you select that one and you want to order it, you can go here and select this guide RNA pair and order it in an all-in-one vector, so you'll get both guides on a single vector. The other way to order it on a 2-vector system, and remember here, if you choose a GFP for this option, it will generate the RFP for the other option automatically. So you can monitor that both plasmids expressing both different guides are in that strain of interest. So this is a convenient way to use our tool and then select the vectors you need. There's 2 ways to use this as well. If you'd like us to clone it, you can select the vectors here and add to cart. If you want to simply make them yourself, you can buy the vectors from us in our Electra-ready cloning method. They're Electra compatible, so they come in our Electra kits. And you can buy the oligos to generate these different guide RNAs and simply make the constructs yourself using this eyeball naked option. So you can simply download the guide RNA sequences along with the necessary overlaps for cloning directly from this link here. So this tool can be used to select guide RNA sequences, or it can be used to enter pre-selected guides into our vectors, or you can just take the guide RNA sequences out and make them yourself after buying the vectors as catalog items from us. So lots of different options for how you can make your targeting constructs. I'm going to go back to the presentation. Again, as I mentioned, this guide RNA tool has nearly instant results. It works both with the wild type and the Nickase Cas9. It is designed to help with mitigating of off-target effects by selecting most unique targets. You can directly order guide RNA in on CRISPR vectors and have them ready for your experiments. If you have any further questions, you can certainly ask us, or also watch our little video on how to use our guide RNA design tool. In addition to this, we also have CRISPR products, as I mentioned, in mammalian cells and certainly in the yeast as well. We're starting to develop things with customers on a case-by-case basis wherever there's interest for other organisms. So what we can do is, for example, codon optimize the Cas9 for your favorite host. We can provide it with different promoters so that you can modulate the expression of Cas9. We can provide versions that allow expression of one or multiple guide RNAs. So we can generate this all-in-one vector. We can also put the reporter protein, like a GFP or RFP, which is also optimized for your host of interest. So there's lots of different things we can do, and we're happy to work with you in your favorite host system. Let me just talk about a couple quick emerging applications, and this is coming close to the end. One is guide RNA libraries. We get a lot of questions about these. These are for functional screens in the range of hundreds to thousands to hundreds of thousands of genes. One could imagine knocking out a set of genes, some family of genes like all kinases in a human genome or such, and they want to screen with a pool of guide RNA targeting vectors, and you could target different genes and look for a certain phenotype. We can generate libraries for single targets. What's novel is we can also generate pairs of libraries. So what I mean by that is libraries of pairs, where if you use the paired nickase strategy, you could still target a given gene using a pair of guide RNAs in a pool where each individual pair is still associated. So you would never get a situation where one half of the nickase pair of one targeting gene is associated with a different gene. These would be pre-associated and would still come in a pool. So this could be useful for targeting pairs of genes in a predetermined manner or targeting genes using this paired guide RNA approach. So if you have any questions about this, this might be a little confusing, but I realize, but definitely come and ask us about it. We can generate libraries of single targets or multiple targets where two targets are pre-associated. We can also this is just by way of closing. We can also offer expression vectors around the lentiviral system, which we have got inquiries for. So if you have interest in using our CRISPR system using lentiviral delivery, do inquire about that. So this is just a conclusion slide. Basically, I described our guide RNA tool. It works both with a double cutter and nickase. We have all-in-one plasmids using Cas9 wild type and nickase with different promoters and different reporters like the GFP and RFP, which can work together. We also offer the ability to express multiple guide RNAs off a single plasmid. We have products around mammalian cells and yeast as we speak. We have E. coli pretty close to being released and other products being developed on a case-by-case basis for any host. Definitely inquire if you have interest in that. And as in terms of emerging applications, we're working on some guide RNA library tools and antiviral delivery. Okay, so that's all I have, and I'll open up for questions now.
Thank you again for joining us today. We hope you've discovered a valuable new set of tools with which to move your research forward quickly and efficiently, and we look forward to working with you on your next project. We'll now answer as many of the questions that remain as possible. And if you have any additional questions, you can always reach the DNA 2.0 team by phone or email. So thank you again, Dr. Patel, for this wonderful introduction to the CRISPR technology. The first most common question was, can you further explain indel frequency and how it's measured?
Yes, sure. Let me back up and go to that slide. So what I mean by that is when you put a double-stranded cut in the DNA, you get a break. And in fact, let me go back to a different slide. So when you put a double-stranded cut in the DNA, it gets repaired commonly, if you don't provide any other cassette, it gets repaired commonly by non-homologous end joining. And what you get is a mutation. If the same sequence comes back together in the correct form, it will just get cut again. So the only ones that should survive are ones that get mutated at that junction. In non-homologous end joining, typically you will put in some mutations commonly, and when that mutation happens, those sites will not get cut again, typically. And that introduces a frame shift or an indel. It can be an insertion of a couple bases or deletion. So that's where we get indels from. And how we measure it is when we do CRISPR, when we target a given site, what we'll do is after expression and after, we'll get single colonies and we'll try to screen individual single colonies by sequencing or by PCR amplifying this region of interest. So this green site here would presumably have been mutated. So we would simply PCR amplify up this region and then sequence to see what happened at this site here. Because we know exactly where the site is, we know exactly where the cut happens, we know exactly where to expect indels or mutations. So that's how we measure indel frequency. It's just a measure of how often do we get back the wild type sequence versus a frame shift.
Thank you. Along that line, someone else just asked very quickly, how efficient is the non-homologous end joining in mammalian cells?
So my understanding, in mammalian cells, it is fairly efficient. We are seeing commonly efficiency of up to 50% in our hands here in the lab. There are reports out there of studies with as many as 5 simultaneous changes in the same organism using CRISPR. So that's pretty remarkable that you can make 5 changes in a markerless fashion by presumably this end joining. So it's fairly efficient in mammalian systems.
Thank you. Another common question that came up was regarding bacterial systems, and do the promoters that we specified work well in bacterial systems, and what do you recommend for that?
That's a good question. We are still in development phase for the bacterial systems. In my experience, expression of Cas9 is not the issue. Expression of gRNA is also probably not the issue based on literature as well. Cutting the DNA in the genome is probably not the problem. It's the downstream repair and things like that. Certainly in E. coli where I've been working. Other bacteria might be different, and if you can supply other helper proteins to help with the downstream recombination or homologous end joining that you're trying to achieve to make your change in the genome, that would help a lot. But I think expression of Cas9 certainly is great in E. coli and probably other bacteria. Expression of GAI should not be a problem either.
Great, thanks. The next question is, what is the maximum size of insertion for new DNA sequences?
That is a good question. It'll depend on your host of interest. It shouldn't be any different from what it is without using CRISPR generally. CRISPR only helps enhance the efficiency, so you have to screen for your clone, then you can do it without markers. But I don't know if it will make a difference to how big you can go. It might a little, because now you can screen for even rarer events than you could before. But it will just depend on the organism of interest and what's been reported about homologous recombination in that system.
Great. Next question. Can you compare the off-target site cleavage frequency between the NickaseNinja and the regular CRISPR vectors?
We can compare, and experiments here are underway. So there's 2 ways to do that. One is you look for predicted off-target sites that are known to be 1, 2, 3 bases off, and you look for what happens there. The other is you just screen the entire genome of an organism by next generation sequencing and see what actually really happened at any site, and compare frequencies of on-target effects or on-target activity versus off-target effects. We have experiments underway, and we're trying to look into that and compare those things.
Great. Another question is on selecting, which is how do you select for knockouts? Can you select positive cells with CRISPR by using antibiotics? What's the actual technique?
To select for knockouts, certainly you can select by using markers if that is your preferred approach. The way CRISPR works, it is certainly for knockouts where you're simply introducing a frame shift and not supplying any recombination per se, it works so efficiently that the whole idea that you do not need to use marker. If you would really prefer to use a marker, you can certainly throw in a marker with the right upstream and downstream homology to replace your gene that you're knocking out with that marker essentially, and that would work just as well. But you do not need to use markers if you don't want to, since if it's as high as 50% of the time that you get indels, or even 10%, as long as you're willing to screen 10 or 20 colonies, you'll find something that's a positive. So it's fairly efficient without the need for markers.
So here's an interesting question. It says, if we are engineering an unconventional organism with a sequenced genome, can DNA 2.0 help us find a suitable promoter?
Certainly, we depending on the organism, of course, we try to borrow from whatever expertise the customer has with that organism, and then we're certainly happy to help with synthesizing and screening up a set of different promoters. Certainly inquire about that, and we're happy to help you.
Great. When using CRISPR in other organisms, such as sac, pichia, algae, etc., what promoters do you use to drive the gRNA expression?
With Saccharomyces cerevisiae, which is what I've been working with most, we've been using these SNR52, they're the SNORNA promoters. They've been published about in the DiCarlo paper from the Church Lab. That's the one I've been using. There are certainly other promoters out there that one can use for these as well.
Great. And I'm just going to say that questions keep coming in, and that's fabulous. We'll get to as many as we can, and those that we can't get to, we will email you replies within the next 24 hours. So the next question is, how long does the homologous recombination overlap sequences need to be?
Again, that homologous recombination sequence length depends on the host of interest. So some hosts like E. coli are not great with recombination on their own. So you need to provide 500 to 1 kb at least of homology on either side. If you are able to do it with the recombinering enzymes or lambda phage enzymes, you can do it with shorter 40 base pair sequences. Again, the way I understand CRISPR, it does not change generally how the recombination mechanism would work or any of that. It just serves as a way to select against anything that is still wild type because those get cut. So you have to still do it like you would with that host without CRISPR. It's just that this greatly enhances the efficiency of it working, so you do not need markers. You could probably get away with maybe slightly shorter arms just because this method is so efficient and you will find rarer events. But it's recommended that you do things just like you would without CRISPR, just that CRISPR will help you get away from using markers.
Good. So clearly another theme that's coming up for a lot of our researchers is additional species. And obviously lots of you are using a variety of species, but a number of questions are coming in about when are we going to add additional species to the gRNA design tool, and what should I do if my species isn't part of that tool yet?
Certainly write to us with the species that you want included in our design tool. We are happy to work with you to get that up. We can, if it's a sequence that you have sequenced and you'd like to keep it private, we can do that too under some circumstances with the right arrangements in place. If it's just a GenBank file that you want to be made available through the tool and that you can use, we're happy to entertain those as well. What we have now is a running list of genomes. We are working on getting those up on our system. There's no barrier to doing that. It's simply just letting us know which ones are of interest to you.
Great. Next question is, do you have the lentiviral plasmids that have CRISPR-Cas9 system in place, since you discussed lentiviral?
I will say that we have some plasmids that we are working with here. If you have your favorite that you like to use, we're happy to work in that system as well. The ones we have are in the process of being validated right now, and we're happy to share that information on a one-on-one basis with you. So definitely inquire about that with us.
Great. A more practical question, how do you sort the cells in which the indel happened out from the wild type cells?
So we usually end up screening for single colonies that have, and then we'll check for whether the mutation is present or not, and we can tell which one it was. So we would simply isolate single colonies and then look for the indels in those single colonies. And if one has it, we have our guy and we keep that then as a positive.
Excellent. This one's an interesting one, and we'll see what we can do with it. Is using gRNA more efficient in activity compared to using crRNA and tracer RNA? Sorry, tacrRNA. Yeah, tracer.
That's a good question. I have only worked with the chimeric single guide RNA because it just intuitively seems like it will be more efficient to me. I have not tried the two RNA system per se, but just intuitively it seems like less components should be better, higher efficiency, less moving parts.
Back to a minutia question, which is how many colonies do you typically check?
Again, it depends on the host and depends on what sort of change I'm putting in and how the guide RNA tool, when I look at the guide RNA, I've selected how well I think it will work. But typically between 4 and 20 clones. 20 is the most I would do before I throw in the towel and try something different.
Okay. Is the transformation done with plasmid stable in the host?
Depends on the system, so in the mammalian system, our plasmid, that's why we have the reporters there so that we can make sure that they're still being expressed. In our hands, certainly 3 to even 7 days we have seen expression of the reporters, so I'd say they're fairly stable through that. They're certainly stable enough to generate the change we need, and then we can get rid of the plasmids as needed. Because after the job is done of editing, you don't want to have the plasmid linger around too long. So it certainly sticks around long enough to do the intended editing in mammalian cells. In the yeast system, we have selection for the plasmid, so you can either leave that selection on or off, and then you would keep the plasmid and retain it, or eventually use it.
Great. I'm going to do 2 more questions, and then we'll wrap things up. So the next question is, can donor DNA be single-stranded rather than double-stranded?
That's a good question. I would say it would depend again on the host, like whether there's a precedent for doing it with single-stranded DNA or not in that host system. I've heard of that done certainly in E. coli and other organisms like that. Mammalian, I'm not 100% sure, but I don't see why not. But I would say check with what has been done in terms of editing by more traditional methods in those hosts, and then that should work just fine with CRISPR as well.
That's good. And then just for a last question, and I know there's lots more, but we'll get to them by email. Do you offer now, or will you be offering in the future, other versions of Cas9 instead of the S pyogenes?
So we operate purely on interest from customers. So if you have such requests, certainly send them our way. We are happy to entertain them. Right now, we do not have any immediate plans of working with other PAMs or other Cas9s from other hosts. But if we receive enough requests for them, we're happy to do it. So certainly contact us. We can talk about that.
Great. Thank you so much, Dr. Patel. Thank you again for joining us today. We hope you've discovered a valuable new set of tools with which to move your research forward quickly and efficiently. We look forward to working with you on your next project. If you have any additional questions, you can contact us by phone or email as noted on this last slide. A copy of the webinar will be available on our website in about 48 hours. And for more information on DNA 2.0 products and services, you can always visit our website. Thank you.